Hello everyone, and welcome. I'm excited to kick off our presentation today. Today, we're diving into the fascinating world of plants with our topic, Anatomy of Flowering Plants. We'll explore the structure, parts, and functions that make these plants so remarkable. So, let's get started and discover what makes flowering plants truly unique. Let's take a closer look at the different types of tissues in plants. Starting with tissues in general, we see that they can be broadly classified into simple tissues and complex tissues. Simple tissues, like parenchyma, collenchyma, and sclerenchyma, are made up of one type of cell and serve various functions such as storage, support, and photosynthesis. Complex tissues, on the other hand, are composed of more than one cell type and work together as a unit. Examples include xylem and phloem, which are part of the vascular tissue system responsible for transport. Focusing on the vascular tissue system, it includes various specialized tissues like xylem and phloem, each with specific roles. Xylem transports water and minerals from roots to leaves, and it includes structures like xylem fibers and tracheids, which provide support and conduction. Phloem transports organic nutrients, especially sugars, throughout the plant, and it contains sieve tube elements and phloem fibers, including phloem parenchyma cells. Now, moving to the stem structures, in monocotyledonous stems, the arrangement of vascular tissues differs from dicotyledonous stems. In dicots, the vascular bundles are arranged in a ring, which is more organized and allows for secondary growth. In monocots, the vascular tissue is more scattered. Let's also consider the tissue systems that make up these structures. The tissue system includes the epidermal tissue system, which covers the plant and provides protection. In leaves, like the dorsiventral or dicotyledonous leaf, the tissue organization is specialized for photosynthesis and gas exchange. The ground tissue system supports this process, with tissues like parenchyma and sclerenchyma providing structure and storage. Finally, the tissue system also includes the meristematic tissues, which are responsible for growth. These are undifferentiated cells that give rise to all other tissue types, enabling the plant to grow in length and girth. Understanding these tissues and their organization helps us appreciate how plants grow, transport substances, and adapt to their environment. A tissue is essentially a group of cells that share a common origin and work together to perform a specific function. Think of it as a team of cells that collaborate to keep the plant healthy and functioning properly. Now, when we talk about tissues, there are two main types we need to remember: meristematic tissues and permanent tissues. These two types differ mainly in their ability to grow and specialize. Meristematic tissues are like the plant's growth zones. They contain cells that are actively dividing, which allows the plant to grow in height and width. On the other hand, permanent tissues are more specialized. They have finished dividing and now perform specific functions like transporting nutrients or providing support. Understanding these two types helps us see how plants grow and maintain their structure. It's a fundamental concept that ties into the overall theme of plant biology and how plants develop and adapt. Now, let's explore the different types of meristems that drive plant growth. First, we have the lateral meristems. These are responsible for the plant's growth in width or diameter, helping the plant become thicker over time. Next, there are the intercalary meristems, which are quite interesting because they allow certain plants, like grasses, to grow from their base. This means they can recover quickly after being cut or grazed. Finally, we have the apical meristems, located at the tips of roots and shoots. These are the primary sites for growth in length, enabling plants to extend upward and downward. Overall, these specialized regions of active cell division, meristems, are essential for the plant's development, allowing it to grow in various directions and adapt to its environment. Now, let's take a closer look at the types of tissues based on their cellular composition. First, we have complex tissues, which are characterized by having many different types of cells working together. These tissues are highly organized and perform specific functions that require diverse cell types. On the other hand, simple tissues consist of cells that are all similar in structure and function. These cells work together in a more uniform way, which makes these tissues somewhat less specialized but still essential for many biological processes. Moving on, we also need to understand that some cells are structurally and functionally specialized to the point where they lose their ability to divide. These are known as permanent tissues. They are crucial for maintaining the stability and integrity of organs and tissues over time. Connecting this to the overall theme, recognizing the differences between simple and complex tissues, as well as the concept of permanent tissues, helps us appreciate how the body organizes its cellular components to perform a wide array of functions efficiently. Now, moving on to slide 6, let's focus on the different types of simple tissues. First, we have sclerenchyma. Sclerenchyma cells are typically thick-walled and provide structural support and strength to the plant. They are often dead at maturity, forming the rigid parts of the plant like the hard shells of nuts or the fibers in the stem. Next, parenchyma. Parenchyma cells are living cells with unevenly thickened walls, giving flexible support to the plant, especially in growing regions like stems and leaf stalks. They help the plant withstand bending without breaking. Lastly, we have parenchyma. Parenchyma cells are the most common type of simple tissue. They are usually thin-walled, living cells that perform various functions such as photosynthesis, storage, and healing. These three types of tissues, sclerenchyma, parenchyma, and parenchyma, are fundamental to the plant's structure and functionality, and understanding them helps us grasp how plants maintain support, flexibility, and perform essential metabolic activities. Cells are isodiametric, which means they tend to be similar in all directions. They can be spherical, oval, round, polygonal, or elongated in shape. This variation in shape helps them perform different functions effectively. Their cell walls are generally thin and are made up of cellulose, providing structural support while remaining flexible. You'll also notice that these cells are either closely packed together or separated by intercellular spaces, which can be important for transport and communication within plant tissues. The primary functions of these cells include photosynthesis, where they produce food for the plant, storage of nutrients, and secretion of various substances. Understanding these cellular characteristics is fundamental to grasping how plant tissues like parenchyma contribute to the overall health and functioning of the plant. Now let's take a closer look at parenchyma, which is situated just below the epidermis in dicotyledonous plants. This tissue is made up of homogeneous layers or patches, providing both strength and flexibility to the plant. The cells in parenchyma are unique because they have thickened corners, mainly reinforced with materials like cellulose, hemicellulose, and pectin. These thickened cell walls are essential for offering mechanical support without sacrificing flexibility, which is crucial for growth and movement. Another interesting point is that parenchyma contains chloroplasts, so sometimes it's referred to as chlorenchyma. This means it can also participate in photosynthesis, especially in young, green parts of the plant. Unlike some other tissues, parenchyma lacks intercellular spaces, which helps it maintain its structural integrity. Overall, parenchyma plays a vital role in supporting the plant's structure and enabling it to withstand various mechanical stresses, connecting well to our broader theme of plant support systems. Now let's look at sclerenchyma, which plays a crucial role in providing mechanical support to plants. These are highly thickened dead cells that can be spherical, oval, or cylindrical in shape. They often have narrow cavities called lumens. You'll find these cells in various plant parts such as the fruit walls of nuts, the pulp of fruits, seed coats of legumes, and even in the leaves of tea plants. Their main function is to give strength and support to the plant structure. Moving on, we have sclerades, which are a type of sclerenchyma. These cells are thick-walled, elongated, and pointed, adding to the hardness and toughness of plant tissues. They contribute significantly to the protective and structural qualities of the plant. Next, let's discuss fibers. These are long, narrow cells with thick, lignified walls that are rich in lignin, making them very sturdy. They contain numerous pits, which are small channels that facilitate the transport of substances. Importantly, fibers are dead cells without protoplasts, meaning they are fully specialized for support rather than metabolic activity. All these types of sclerenchyma, sclerades, and fibers, are essential for maintaining the plant's structural integrity, especially in parts that need to withstand physical stresses. Their thickened walls and dead state make them ideal for providing durable support throughout the plant's life. Now, let's talk about complex tissues. These are made of more than one type of cell that work together as a unit. This collaboration allows the tissue to perform specific functions more efficiently. In plants, for example, two main types of complex tissues are xylem and phloem. Xylem is responsible for transporting water and minerals from the roots up through the plant, while phloem carries the food made in the leaves to other parts of the plant. Recognizing these different cell types working together helps us understand how plants stay healthy and grow. It's a great example of how diversity within tissues contributes to the overall function of the organism. Now, let's take a closer look at the different components of the xylem, which is essential for water and mineral transport. Starting with the xylem parenchyma, these are living cells that help in storage and lateral transport. Moving on, we have vessels, which are the main conducting elements in many plants, especially in angiosperms, allowing efficient water flow. Next are xylem fibers, which provide structural support and mechanical strength to the plant. Then we have tracheids, which are also conducting cells found mainly in conifers and other gymnosperms, helping in water conduction and support. Altogether, these four types of elements, parenchyma, vessels, fibers, and tracheids, work together to ensure the xylem performs its vital role. This tissue not only transports water and minerals from the roots to the stem and leaves, but also reinforces the plant's structure, making it resilient. Understanding these components helps us appreciate how plants maintain their hydration and stability. Now, let's take a closer look at the inner layers of the cell walls, which have some interesting thickening patterns. These thickenings are not uniform. In fact, they vary quite a bit in form, which plays a crucial role in the cell's function and strength. Moving on to tracheids, these are specialized cells essential for water conduction in plants. They are elongated, tube-like cells that help transport water from roots to leaves. The thickening of their cell walls provides structural support, preventing collapse under the pressure of water movement. This variation in wall thickening is key to understanding how plants maintain both flexibility and strength in their vascular tissues. So, by studying these differences, we gain insight into how plants adapt their internal structures to optimize function and resilience. Let's now turn our attention to vessels, which are a crucial part of our circulatory system. Vessels are the channels through which blood travels throughout our body, and they come in different types, each with a specific role. Arteries carry oxygen-rich blood away from the heart to the tissues, and they need to be strong and elastic to handle high pressure. Veins, on the other hand, return deoxygenated blood back to the heart, and they often have valves to prevent backflow. Capillaries are the smallest vessels, acting as the sites of exchange between blood and tissues. Understanding how these vessels work together helps us see the bigger picture of how nutrients, oxygen, and waste are transported in our bodies. This interconnected system is fundamental to maintaining overall health and function, and it ties directly into the themes we've discussed about circulation and homeostasis. Now, let's turn our attention to xylem fibers. These are specialized cells found within the xylem tissue, and they play a crucial role in providing mechanical support to the plant. Unlike the vessels that are primarily responsible for water transport, xylem fibers are more about strength and stability. They are long, slender cells with thick cell walls made of lignin, which gives them rigidity. You can think of them as the plant's internal scaffolding, helping the plant stand tall and withstand various stresses like wind or the weight of the leaves and branches. Their presence is essential for maintaining the overall structural integrity of the plant, especially in woody plants that grow quite large. So, in the bigger picture of plant transport systems, while xylem vessels handle water conduction, the fibers ensure the plant remains upright and resilient. Understanding these fibers helps us appreciate how plants balance their need for transport with their need for support, which is vital for their survival and growth. All right, let's take a closer look at this slide. Here, we're seeing some key terms related to plant tissue organization. First, you'll notice the labels exarch and endarch. These refer to the types of xylem development, which is essential for water conduction in plants. An exarch xylem develops from the outside inward, meaning the earliest formed xylem is on the outer side, while endarch develops from the inside outward, with the earliest xylem on the inner side. Next, we have xylem parenchyma. This is a type of ground tissue found within the xylem. It plays important roles, such as storing nutrients and aiding in the repair and maintenance of the xylem tissue. It's a supportive and versatile tissue that helps maintain the functionality of the vascular system. Connecting this to our overall theme, understanding these different types of xylem development and associated tissues helps us appreciate how plants adapt their vascular systems for efficient water transport and structural support. Recognizing these distinctions is fundamental to grasping how plants grow and respond to their environments. Now, let's talk about how the phloem functions in plants. Its main role is to transport food materials, mainly the sugars produced in the leaves, to other parts of the plant that need energy or stored for later use. This process is essential for the overall growth and development of the plant. The phloem is composed of several key components. First, we have the sieve tube elements, which are the main conduits for transporting the sugars. These are specialized cells that form long, tube-like structures. Alongside them are the companion cells, which support the sieve tubes by helping with the loading and unloading of nutrients. Next, there's the phloem parenchyma, which plays a role in storage and lateral transport within the phloem tissue. Finally, the phloem fibers provide structural support, ensuring that the phloem tissue remains intact and functional as it carries out its transport duties. Connecting this to the overall theme, understanding the structure of the phloem helps us appreciate how plants efficiently distribute the energy they produce, supporting their growth, reproduction, and response to the environment. Now, let's take a closer look at the sieve tube elements. These are long, tube-like cells that run lengthwise through the plant's phloem. They're arranged in a way that allows for efficient transport, and you'll notice that their end walls are perforated to form what we call sieve plates. These plates act like gateways, facilitating the flow of nutrients between the cells. What's interesting is that these cells contain cytoplasm and a large vacuole, but notably, they lack a nucleus. This might seem unusual, but it actually helps optimize the transport process. However, controlling the functions of these sieve tube elements is the nucleus of their companion cells. These companion cells are closely associated with the sieve tubes and play a crucial role in managing activities like loading and unloading sugars. So, in essence, the sieve tube elements are specialized for transport, and their activities are coordinated by the companion cells, ensuring the efficient movement of nutrients throughout the plant. This teamwork is a perfect example of how plant cells work together to sustain life. Now, let's focus on the specialized parenchymatous cells involved in the phloem. These include sieve tube elements and companion cells, which work closely together. They are connected through structures called pit fields. These pit fields are crucial because they facilitate communication and transport between the cells. This connection helps maintain the pressure gradient within the sieve tubes, which is essential for efficient nutrient transport. Essentially, these specialized cells and their connections ensure that the flow of nutrients, like sugars, is smooth and effective throughout the plant. This coordination is a key part of how plants distribute energy and sustain their growth. Now, let's take a closer look at phloem parenchyma. These cells are made up of elongated, tapering cylindrical cells that have dense cytoplasm and a nucleus. They play a crucial role in the storage and transport of various substances within the plant. You'll notice that these cells contain cellulose and have pits, which are essential because they allow for plasmosomal connections between neighboring cells. These connections facilitate communication and transport of materials across cells. Phloem parenchyma is particularly important because it stores food, resins, latex, and mucilage, acting as a reserve for the plant. An interesting point to remember is that these cells are absent in monocotyledons, which highlights some of the structural differences between different plant groups. Overall, Phloem parenchyma contributes significantly to the plant's ability to store and move vital substances, supporting the overall health and functionality of the plant. Now let's take a closer look at the phloem fibers, also known as blast fibers. These fibers are made up of sclerenchymatous cells, which are known for their thick, lignified walls. Interestingly, they are absent in the primary phloem, meaning they develop later in the plant's growth. These fibers are elongated and unbranched, with pointed, needle-like tips that help them provide strength and support. At maturity, they lose their protoplasm and become dead, which is typical for sclerenchymatous fibers, as this dead tissue provides rigidity. You'll find these fibers in commercially important plants like jute, flax, and hemp, where they are used widely in industries for making textiles, ropes, and other durable materials. Overall, these fibers are crucial for the structural integrity of the plant and have significant economic value due to their strength and versatility. Let's take a closer look at the structure of the phloem. First, you'll notice that sieve tubes are larger in size. This size difference is important because it relates to their function in transporting nutrients. Now, within the phloem, we have two main types, the metaphloem and the protophloem. The metaphloem, which is the outer part, contains the sieve tubes that are larger and responsible for the main transport of food. On the other hand, the protophloem, which is located more towards the interior, has narrower sieve tubes. These narrower tubes are typically involved in the early stages of development and support. So, understanding these structural differences helps us appreciate how the phloem efficiently moves nutrients throughout the plant. The size variation between the sieve tubes actually reflects their specific roles in the overall transport system. Now let's take a closer look at the different tissue systems in plants. First, we have the vascular or conducting tissue system. This system is responsible for the transport of water, nutrients, and sugars throughout the plant. It includes structures like the xylem and phloem, which work together to ensure the plant's survival and growth. Next, there's the ground or fundamental tissue system. This is the most abundant tissue type and plays several roles, including support, storage, and photosynthesis. It forms the bulk of the plant's structure and helps in maintaining overall plant health. Then we come to the epidermal tissue system. This is the outermost layer of cells that covers the plant. It acts as a protective barrier against physical damage and pathogens, and it also helps minimize water loss through specialized structures like stomata. When we look at these systems based on their structure and location, it helps us understand how plants are organized internally to carry out their vital functions. Each system has a distinct role, but they all work together harmoniously to support the plant's life processes. This interconnectedness is really fascinating and underscores the complexity of plant biology. Let's take a closer look at the epidermal appendages, specifically trichomes and hairs. Hairs on plants, particularly root hairs, are unicellular elongations of epidermal cells that play a crucial role in absorption. They increase the surface area, making nutrient uptake more efficient. On the other hand, trichomes are multicellular structures that can be either branched or unbranched. They can be soft or stiff and often have secretory functions. An important role of trichomes is to prevent water loss through transpiration, acting as a protective barrier. Now, moving on to stomata, which are specialized structures found in the epidermis of leaves. These are essential for regulating transpiration and gaseous exchange. They open and close to control water vapor and gases like oxygen and carbon dioxide, maintaining the plant's internal balance. Outside the epidermis, you'll notice a waxy layer called the cuticle. This thick, protective layer prevents water loss, especially in leaves. Interestingly, this cuticle is absent in roots, allowing them to absorb water more effectively. The outermost layer of the epidermis is called the epidermis itself, made up of elongated, tightly packed cells that form a continuous single layer. This layer acts as a protective covering for the entire plant body. In summary, the epidermis, along with its appendages like hairs, trichomes, and structures like stomata, plays a vital role in protecting the plant and regulating its interactions with the environment. All right, let's talk about the ground tissue system. This part of the plant primarily consists of simple tissues, which means they are made up of only one type of cell. Importantly, these tissues exclude the epidermal layer and the vascular bundles, so we're focusing on the internal supportive and metabolic tissues. One of the key components here is the mesophyll, which you might remember is crucial for photosynthesis. It contains the chlorenchyma cells that trap sunlight and convert it into energy. Overall, the ground tissue system plays a vital role in supporting the plant's structure and facilitating vital processes like photosynthesis, making it an essential part of the plant's overall functioning. Now, moving on to the vascular tissue system, let's take a closer look at the types of vascular bundles. There are primarily two types, the radial and the conjoint. In these bundles, xylem and phloem are together, forming a crucial part of the plant's transport system. The radial type is often seen in certain roots, where the xylem and phloem are arranged alternately in a radial fashion. On the other hand, the conjoint type is more common in stems and includes both xylem and phloem together within the same bundle. Understanding these differences helps us appreciate how plants adapt their vascular arrangements based on their structure and function, which ties back to the overall theme of how plants efficiently transport water, nutrients, and food throughout their bodies. Now, let's look at the structure of the dicotyledonous stem. The pericycle, which is the outermost layer of the vascular cylinder, forms semilunar patches of sclerenchyma. These patches provide mechanical support and protection. Moving inward, you'll notice that the vascular bundles are arranged in a ring pattern. These bundles are conjoint, meaning they contain both xylem and phloem, and they are open, allowing for secondary growth. They are also endark, which indicates that the protoxylem develops towards the center, a characteristic feature of dicots. Surrounding these bundles is the pith, which is primarily composed of parenchyma cells and functions in storage. Next, the endodermis is a key layer. It's rich in starch grains, forming what we call the starch sheath. This layer plays an important role in regulating the movement of substances into the vascular tissue. The cortical layer, lying outside the endodermis, is divided into 3 subzones, which we'll explore further as we analyze the overall structure. The hypodermis is just beneath the epidermis, providing additional protection and support. Now, speaking of the epidermis itself, it consists of a cuticle to prevent water loss, trichomes, which are hair-like structures that can serve various functions, and stomata that facilitate gas exchange. Finally, the cortex is divided into these 3 subzones, each with specific roles, contributing to the overall function and integrity of the stem. Understanding this layered organization helps us appreciate how dicot stems support their growth and respond to their environment. Now, let's look at the structure of the monocotyledonous stem. It is characterized by a sclerenchymatous hypodermis, which provides additional support and protection to the stem. Scattered vascular bundles are embedded within the ground tissue, and unlike dicots, these bundles are surrounded by a bundle sheath. The vascular bundles themselves are conjoint, meaning both xylem and phloem are together in the same bundle, and they are closed, which is typical for monocots. They are also endark, indicating that the xylem develops towards the center of the stem. Additionally, you'll notice that the peripheral vascular bundles are smaller compared to those at the center. Interestingly, the phloem parenchyma, which is often seen in dicots, is absent here. Instead, some vascular bundles contain water-containing cavities, which can play a role in tissue hydration and flexibility. Understanding these features helps us see how monocots are adapted structurally to their environment, supporting the overall theme of plant structural diversity and specialization. Now, let's look at how the leaf structure is organized. The epidermis, which we've seen before, covers both the upper and lower surfaces of the leaf. It acts as a protective barrier, preventing water loss and offering some defense against pathogens. Between these 2 epidermal layers lies the mesophyll, which is the main tissue responsible for photosynthesis. It's where the magic of converting light into energy happens. Moving on, the vascular system is integrated within the leaf, providing essential transport of water, nutrients, and sugars. The lamina, or the leaf blade, has three main parts, which are crucial for understanding how the leaf functions as a whole. In the context of a dorsiventral, or dicotyledonous, leaf, these structures work together efficiently. This layered organization not only supports the leaf's role in photosynthesis, but also highlights the intricate design that allows plants to thrive. Remember, understanding these components helps us appreciate the complexity and elegance of plant biology as a whole.